Cells cannot live forever. In fact, most cells in the body only live for a few days or weeks. New cells must continually be formed. DNA must be replicated in order for new cells to form, as most cells form in a process called mitosis. In mitosis, the chromosomes in one cell must separate into two new cells, and each cell needs separate DNA strands found within each chromosome. It takes approximately an hour to replicate a section of a DNA strand, or 50 base pairs each second. Cells need to copy their DNA when they divide. This process is called DNA replication. To understand how DNA is copied, we first need to understand the structure of DNA. DNA is made of two long strands that wrap around each other to make the double helix. Each strand is composed of a sequence of molecules called nucleotides. Bases are a part of the nucleotides. There are four possible bases: adenine, thymine, cytosine, and guanine. The bases of one strand pair up with the bases of the other strand in a very simple way called the base pairing rule. C pairs up with G, and A pairs up with T. What this means is that the DNA sequence of one strand completely defines the DNA sequence of the other strand. An easy way to remember this base pairing rule is to remember that C and G are curvy characters, which A and T are built with linear sides. Because of how the base pairs are set up, the two strands come together like a molecular zipper. During replication, the strands are unzipped and each is copied following the base pairing rule. So, if there is an A, when you make the copy, you should put in a T and vice versa. The same holds true for G and C. When replication is complete, each new DNA contains one old and one new strand, or one parental and one daughter strand. Every step of DNA replication needs enzymes to do the work. As we go through this, remember, all that is happening is the DNA is being unzipped and each side of the zipper is being copied. The first step in DNA replication is to separate or unzip the two strands of the double helix. The enzyme in charge of this is called a helicase because it unwinds the helix. The point where the double helix is opened up and the DNA is copied is called a replication fork. Once the strands are separated, an enzyme called DNA polymerase copies each strand using the base pairing rule. The two strands are not exactly copied the same way. Because the polymerase can only work along the strand in one direction, 5 side to 3 side, it uses a slightly different strategy to copy the DNA on each strand, the leading and lagging strand. DNA polymerases are not doing all of this on their own. Many more enzymes help out. For example, there are some enzymes that stitch up the newly made strands. Some enzymes prime the DNA so that the polymerases can start copying. Other enzymes remove these priming sites and replace them with proper DNA. Okazaki fragments are short, newly synthesized DNA fragments that are formed on the lagging template strand during DNA replication. They are complementary to the lagging template strands, together forming short double-stranded DNA sections. Although synthesis of the lagging strand involves only half the DNA in the nucleus, it is twice as difficult to process Okazaki fragments than it is to synthesize the leading strand. During this process, RNA and DNA primers are removed, allowing the Okazaki fragments to attach to the lagging DNA strand. While this process seems quite simple and repetitive, defects in Okazaki fragment maturation can cause DNA strand breakage, which can cause varying forms of chromosome aberrations. Several defects of Okazaki fragment maturation may halt DNA replication and even induce cell death. However, while subtle defects do not affect growth, they do result in future varying forms of genome instabilities. Let's review what we've learned. 1. The enzyme DNA helicase unzips the DNA double helix. 2. In DNA replication, mutations can sometimes occur when the wrong nucleotide is added. DNA polymerase can backtrack and remove the wrong nucleotide and add the right one. This makes it able to proofread its work. 3. When DNA polymerase is done, 2 identical strands of DNA have been formed, each containing one old strand and one new strand. The nucleotide sequences in both DNA molecules are identical to each other and to the original DNA molecule.